I want you to think about a scar, maybe one you have on your body right now.
Yeah, like from a childhood fall or maybe a surgery.
Exactly. If you press on it, you can feel that it's fundamentally different from the skin around it, right?
Right. It's dense.
It's tough. It just doesn't have that natural yield. Like imagine squishing a soft marshmallow versus squeezing a hard, dense rubber ball.
That's a great way to picture it.
Now, I want you to imagine taking that rigid, unyielding rubber ball environment and putting it deep inside your body, surrounding your delicate internal cells. Today, we are going into a fascinating and honestly kind of unsettling deep dive.
It really is. We're looking at this groundbreaking 2026 paper from the journal Cancer Cell by Hayward and colleagues.
And our mission today is to understand a massive, a complete paradigm shift in biology. How does the sheer physical stiffness of human tissue actively cause genetic mutations?
Yeah, it's a complete reframing of how we view the origins of disease, because for a long time, clinical data has shown us that cancer frequently arises in fibrotic tissue.
Which is essentially just internal scar tissue.
Right, exactly. If you have excessive stiff collagen buildup in an organ, your cancer risk just skyrockets. But, the prevailing assumption in medicine was always that this stiffness just provided like a protective fortress for tumors.
Like the tumor was already there, and the scar tissue just kept the immune system out.
Or maybe a fertile soil that a random cancer mutation happened to fall into. But this new research completely dismantles that. It shows us the precise microscopic mechanism of how physical tension alone acts as a mutagen.
It acts as the spark itself.
Yes. Today, we're going to explore how physical forces actually confuse your immune system, and how that confusion literally edits and breaks your DNA.
It sounds almost like science fiction, that physics can rewrite genetics. But before we get down to the molecular level, we need to understand the macro evidence. The crime scene, so to speak.
The crime scene, yeah.
What made these researchers even suspect that pushing on a cell could scramble its genetic code?
Well, the initial clue came from a massive human data set. It's called the Cancer Genome Atlas, or TCGA. The researchers were analyzing breast cancer patients, and they isolated a specific variable, the fibrosis signature.
Okay, so they wanted to see if the amount of dense, cross-linked collagen in a tumor correlated with anything else.
Exactly. And they found that patients with a highly fibrotic tumor environment had significantly worse overall survival rates.
Which, that wasn't too surprising on its own.
Right. We kind of knew that. But the crucial finding, the really wild part, was that these same patients had a massive spike in their tumor mutational burden, or TMB.
So their tumors were carrying way more genetic errors, like a higher volume of typos in their DNA blueprint.
Precisely. The denser the scar tissue, the more mutated the cancer. But, a correlation in a database doesn't prove that the stiffness is the actual culprit.
Right. Correlation doesn't equal causation.
Exactly. To prove that, you have to actually measure the physical tension of the tissue and watch what happens to the cells inside it. So they used a physical measurement technique called atomic force microscopy, or AFM.
Atomic force microscopy. Okay. How does that work?
If you want a visual, imagine a microscopic record player needle.
Oh, interesting.
Yeah. This tiny, incredibly sensitive cantilever physically pushes down onto the tumor stroma, the supportive connective tissue, and measures exactly how much that tissue pushes back.
And that resistance gives them a hard physical number for the stiffness.
Yes. It calculates a measurement of elasticity called Young's modulus. And they didn't just, poke a few random spots. They paired this physical probing with an AI tool.
Of course, there's always an AI tool, right?
Right. It's called StiffMap. So StiffMap analyzes the visual structure of the collagen and accurately maps out the stiffness across the entire tissue landscape.
Okay, so they have a highly detailed topographical map of physical stiffness.
And when they overlaid that physical stiffness map with a biological map of the cells, they found something undeniable. Wherever the tissue was the absolute stiffest, the neighboring epithelial cells were overflowing with a specific marker.
And this marker is called gamma H2AX.
Gamma H2AX, yes.
Okay, let's make sure we clearly understand the stakes here.
Yes.
Because gamma H2AX isn't just some random protein floating around.
No, it's definitely not.
If we picture the DNA helix like a long, twisted zipper, it's constantly unzipping to read instructions and zipping back up. Now, a normal cell can handle minor damages to, say, one side of the zipper.
Sure, single-strand breaks happen all the time.
But gamma H2AX is the cellular alarm bell that rings when both sides of the zipper are violently snapped in half.
Exactly.
It's the universal marker for a double-strand break, which is, It's one of those catastrophic things that can happen to a cell's blueprint.
That's the perfect way to visualize it. It's a red flashing light indicating a full structural failure of the DNA. And these alarms were flashing wildly right in the areas of maximum physical tension.
But I have to stop you here because mechanically there is a missing link.
Okay, what do you mean?
Well, a cell is largely made of water. It's squishy. If I have a water balloon with a zipper floating inside it and I squeeze the outside of the balloon, the zipper inside doesn't just snap in half.
Right, the physics don't directly translate like that.
Exactly. The physical pressure of the stiff tissue has to be translated into a chemical attack inside the nucleus. So who is the biological middleman? How does a squeeze turn into a broken zipper?
You're touching on the exact gap the researchers had to cross. And to find that middleman, we have to look at something called mechanotransduction.
Mechanotransduction. So how a cell feels its environment.
Yes. When the extracellular tissue hardens, the cell doesn't just passively get squished. It actively feels the tension through receptors on its surface, and it tries to brace itself.
Bracing itself how?
By pulling hard on its own internal structural scaffolding, the cytoskeleton. This involves a mechanical pathway called ROCK-dependent actomyosin contractility.
So it's basically flexing its internal muscles to push back against the hard wall.
Exactly. And that immense physical strain triggers a panic response. The mechanical pulling, combined with an inflammatory pathway involving IL-6 and JAK1 signaling, activates a specific transcription factor.
And this transcription factor is STAT3.
STAT3, yeah. You can think of STAT3 as the cell's ultimate SOS panic button. When tension triggers STAT3, it moves right into the cell's nucleus and forces the cell to start pumping out a cocktail of inflammatory chemicals.
So the physical squeeze forces the cell to shoot up a chemical flare.
A bunch of flares, actually. Specific chemokines and cytokines like CCL3, CCL5, CCL12, and CSF1.
Okay, that's a lot of acronyms, but basically they are chemical beacons. Who are these beacons trying to signal?
They're calling for the heavy-duty cleanup crew of the immune system, the macrophages, specifically tumor-associated macrophages, or TAMs. These chemicals lure them out of the bloodstream and directly into this stiff, fibrotic tissue.
Okay, I'm following.
And the researchers proved this sequence beautifully using a specialized mouse model for breast cancer, the PIMT mouse. They administered a drug, a LOX inhibitor called BAPN, which basically softens the tissue.
So the drug prevented the scar tissue from hardening.
Right. And if the tissue stays soft,
the cell never feels the squeeze, it never hits the STAT3 panic button, and it never shoots up the flare.
The results were striking. By keeping the tissue soft, macrophage recruitment just plummeted. And most importantly, those flashing gamma H2AX alarms the double-strand DNA breaks virtually disappeared.
Wow.
Yeah. And they confirmed this again by genetically modifying a mouse to lack that epithelial STAT3 alarm entirely, a STAT3 knockout. Without the alarm, no macrophages arrived, and the DNA remained perfectly intact, regardless of how stiff the environment was.
Okay, let's pause and do a quick recap for the listener, just to lock this in. 3 bullet points.
What for?
Number 1, the tissue gets physically stiff and dense. Number 2, the cells panic from the mechanical stress and activate the STAT3 alarm. And number 3, that alarm chemically recruits macrophages to the scene.
Perfect summary.
And I want you, the listener, to think about a time you pulled a muscle. The tissue became stiff, swollen, and inflamed. What if that physical stiffness was actually sending out chemical beacons to your immune system?
It's happening in your body more often than you think.
But here is the massive contradiction we need to address. Macrophages are supposed to be the good guys.
Right, they're the paramedics.
They clear out dead cells, they fight off infections. Why would summoning the paramedics result in the cell's DNA getting snapped in half?
Because the paramedics are stepping into an environment they just aren't equipped for. You see, macrophages are themselves highly mechanosensitive.
Meaning they also feel the physical stiffness.
Exactly. When they enter soft, healthy tissue, they go about their normal cleanup duties. But when they are pulled into a highly rigid, high-tension fibrotic matrix, they essentially undergo a massive sensory overload.
They panic.
They panic. The sheer physical stretching of their own cell membranes activates an enzyme complex inside them called NADPH oxidase, or NOx.
NOx.
And in this incredibly stiff environment, NOx gets locked into absolute overdrive.
And what exactly does NOx do when it's in overdrive?
NOx is a generator for reactive oxygen species, or ROS. These are highly unstable molecules. They desperately want to steal electrons from anything they touch.
So it's oxidative stress.
Massive oxidative stress. Usually, a macrophage uses a quick, tiny burst of ROS, like a little chemical blowtorch, to destroy an invading deuterium.
Right.
But in this scenario, there are no bacteria. It's just the constant, relentless physical squeezing of the stiff tissue, causing the macrophage to fire its chemical blowtorches continuously.
It's almost like a hazmat team being called into a building. But the hallways are so incredibly narrow and cramped that they start to panic.
I like this analogy. Keep going.
So they turn on their decontamination blowtorches, but because they are squeezed tight against the walls, the blowtorches just bounce back and start melting their own hazmat suits.
That is a brilliant and honestly horrifyingly accurate analogy, because the macrophage's own cell membrane is made of lipids, specifically polyunsaturated fatty acids, or PUFAs.
PUFA.
The massive amount of ROS generated by the panicking NOx enzyme violently attacks those delicate lipids. It's a process called lipid peroxidation. The macrophage is literally rusting from the inside out.
Wait, cellular rust?
Basically, yeah. It's converting its healthy cell membrane into a toxic sludge of oxidized phospholipids, or OXPLs.
Okay, I follow the hazmat suit analogy, but this brings up another logical hurdle.
Okay, hit me.
If the macrophage is melting its own suit, if the oxidized lipid rust is inside the macrophage, how does that damage reach the epithelial cell next door?
That is the million-dollar question.
Because the DNA is locked safely away in the nucleus of a completely separate cell. So how does it cross over?
That was the next massive puzzle the team had to solve. And the secret lies in what happens to those oxidized phospholipids as they break down. They don't just sit there in the macrophage.
Okay, what do?
They degrade into highly reactive, diffusible lipid aldehydes, specifically molecules like MDA, acrolein, and the most notorious one in this study, HNE.
HNE. Let's use a little memory trick for the listener here.
Yeah.
Let's call HNE a highly nasty electrophile.
Highly nasty electrophile. I love that because that's exactly how it behaves chemically. These aldehydes are small and they are highly hydrophobic.
Meaning they hate water, so they love fat.
Exactly. They slip right through fatty cell membranes with zero resistance. They don't need a special transport channel or a receptor. The HNE simply diffuses out of the burning macrophage, drifts across the microscopic gap between the cells, slips right through the membrane of the neighboring epithelial cell, and penetrates straight into its nucleus.
Oh, wow. So the aldehydes are literally the toxic chemical smoke billowing out from the burning hazmat suit. And that smoke drifts right through the open windows of the neighboring house.
Yes, and once that smoke is inside the nucleus, it chemically binds directly to the DNA helix. It creates what biologists call bulky adducts.
Bulky adduct.
Yeah, if we go back to our zipper analogy, imagine shoving a giant hardened wad of chewing gum right into the teeth of the zipper. It physically warps and distorts the shape of the genetic code.
But our cells aren't totally defenseless against this, right? I mean, we have repair crews constantly checking the DNA for damage.
We do. The primary defense against bulky adducts is the nucleotide excision repair pathway, or NER.
NER.
It acts as a molecular scissor. It tries to cut out the gum and patch the hole. It also tries to fix the double-strand breaks. But remember the context that the tissue is permanently stiff.
Which means the macrophages are permanently panicking.
Which means the aldehyde smoke is constantly pouring in, the repair system gets completely, hopelessly overwhelmed.
It's trying to repair a delicate ancient manuscript while the library is actively on fire.
Exactly. And when the cell tries to urgently cut and patch hundreds of these sites simultaneously, it makes sloppy mistakes. We see frame-shift indels where random genetic letters are inserted or deleted.
Just total chaos in the genetic code.
And transversion mutations, like T to G substitutions, where the wrong base pair is swapped in. And this isn't just harmless background noise. The researchers prove that this specific aldehyde damage frequently knocks out crucial tumor suppressor genes.
Like which ones?
Like CAMT2D, which is a vital gene that normally protects the body against cancer metastasis.
Okay, let's stop and synthesize this whole sequence, because understanding this chain reaction is the absolute key to the entire deep dive.
Let's do it.
4 bullet points this time. Number 1, stiff tissue makes macrophages overproduce ROS. Number 2, that ROS burns their own lipids, turning them into oxidized phospholipids, or OXPLs.
Right.
Number 3, those OXPLs degrade into floating aldehydes, that toxic HNE smoke. And number 4, the aldehydes drift into neighboring cells, gum up the DNA zipper, and force catastrophic sloppy genetic repairs.
That is the complete molecular blueprint from physical tension to genetic mutation.
We've officially connected the physical push to the broken genetic code. But, In science, mapping out a theory in a petri dish is only half the battle.
It's really just the beginning.
The ultimate test is whether this exact chain reaction dictates cancer progression in a living, breathing organism, and more importantly, whether we can stop it.
Right, because if this is really driving the mutations, then intervening anywhere in that chain should alter the course of the disease.
So how do they test this in living models?
They engineered two incredibly elegant scenarios in mice. One to make the aldehyde smoke drastically worse, and one to completely neutralize it.
Okay, let's hear the worst one first.
So first, they looked at a natural cellular defense mechanism called GPX4. GPX4 is essentially the antioxidant fire extinguisher that normally cleans up those oxidized lipids.
Okay.
They created a mouse model where the macrophages completely lack this GPX4 gene.
So they basically sent the hazmat team in but took away all their fire extinguishers.
Exactly, and the results were devastating. Without that defense, the macrophages accumulated astronomical levels of oxidized lipids.
The rust just took over.
The toxic H&E smoke skyrocketed, the gamma H2AX DNA damage in the surrounding tissue spiked, and these mice developed incredibly aggressive, rapidly metastasizing tumors. They proved that if you let the lipid rust run wild, the cancer accelerates.
Which is terrifying, obviously, but it perfectly validates the mechanism.
It does.
So what about the other side of the coin? Can we intercept the smoke and make it better?
This is where the research becomes incredibly hopeful. They used a specialized transgenic mouse model that produces a unique antibody called EO6. EO6 SEFV, technically.
And what does EO6 do?
Think of EO6 as a molecular sponge. It specifically seeks out and binds to oxidized phospholipids. It neutralizes the rusting lipids before they can break down into the toxic aldehyde smoke.
Oh, that's brilliant. So the macrophages might still panic from the stiffness, but the EO6 sponge soaks up the damage before it can drift next door.
Exactly. And in the EO6 mice, the toxic H&E levels absolutely plummeted. The gamma H2AX alarms went quiet, meaning the DNA was protected. The overall tumor burden was profoundly reduced.
Just by soaking up the smoke.
Yeah. And they even achieved similar protective results by treating cells in a dish with a simple supplement called L-carnosine.
L-carnosine?
Yeah. It acts as a direct scavenger for the aldehydes. It sweeps them up.
This is, it's mind-blowing when we were talking about treating tumors, but I want to push back on the timeline here for a second.
Sure, go ahead.
If the physical stiffness causes the mutations, does that mean the DNA damage is happening before a person actually gets cancer?
That question leads us to perhaps the most vital clinical application of this entire paper. To figure that out, the researchers looked at a very real-world human condition, mammographic density.
Okay, mammographic density, MD.
When women get mammograms, doctors assess how dense the breast tissue is. High density means there is more fibrotic glandular tissue and less fat.
And high breast density isn't just a scanning issue, it's a known major risk factor for developing breast cancer.
It is a massive risk factor. Women with high mammographic density have much stiffer breast tissue and up to a 4-fold higher lifetime risk of breast cancer.
A 4-fold risk just from having denser tissue.
Yeah. So the researchers wanted to know is this exact physical tension pathway, the hidden reason behind that risk. So they analyzed breast tissue from women with high density who had chosen to undergo preventative prophylactic mastectomies.
So just to be crystal clear for the listener, this is tissue from healthy women. There are absolutely no cancer cells present in this tissue at the time of surgery.
None. Histologically, if you look at it under a standard microscope, it is completely normal, cancer-free tissue.
Okay.
But when they looked at the molecular level, looking for our markers, it was a disaster zone.
You're kidding.
No. In this dense, stiff tissue, they found massive swarms of recruited macrophages. They found significantly elevated levels of the toxic H&E smoke, and most shockingly, the otherwise healthy epithelial cells had dangerously high levels of gamma H2AX.
Wait, really? The double-strand breaks were already happening?
They were already happening.
Wow. So because their natural tissue architecture is physically stiffer, it is constantly recruiting these confused immune cells, which are constantly baiting their healthy DNA in toxic smoke.
Exactly.
The physical stiffness is actively preloading the genetic gun years before a tumor ever forms.
You've captured the gravity of it perfectly. The stiffness creates a chronically inflamed environment where the DNA is under constant siege. It accumulates mutations over years and years, and statistically, it's really only a matter of time before one of those sloppy repairs hits a critical driver gene and the cancer officially begins.
This radically alters how I think about disease prevention. I mean, we've spent decades trying to fight cancer by targeting the mutated cells after they appear. We search for the genetic typo. But this research is telling us that for high-risk individuals, the genetics are downstream of the physics.
That is the paradigm shift. If we know that high mammographic density or, other fibrotic conditions are actively mutating DNA through this aldehyde pathway, we have entirely new targets for prevention.
Right. What if, instead of just watching and waiting, we could design prophylactic therapies?
Exactly. Therapies that scavenge lipid aldehydes, kind of like the L-carnosine did in the lab. Or therapies that utilize antibodies like EO6 to neutralize the oxidized lipids early on.
We could theoretically intercept the chemical smoke and stop the mutagenic process before the crucial cancer-causing error is ever written.
We aren't just fighting the genetic code anymore. We are fighting the biophysics of the tissue itself.
That is incredible. And that brings me to a final lingering thought for you, the listener, to take away from this deep dive.
Let's hear it.
We've focused heavily on breast cancer and mammographic density today because that's where the data is. But think about the rest of the human experience.
Yeah, stiffness happens everywhere.
Right. As we age, our tissues naturally lose their elasticity and become stiffer. Think about the scar tissue that forms in our lungs after severe respiratory infections. Or think about the highly fibrotic, rigid tissue that develops in the liver or around the heart as a result of obesity or metabolic syndrome.
It's all the same physical trigger.
If sheer physical stiffness can drive genetic damage through confused immune cells, could keeping our tissues physically soft, pliable, and free of chronic inflammation literally be the key to protecting our DNA from the inside out?
It's a huge question.
Are the physical forces of our bodies quietly writing our genetic destinies every single day? Next time you feel a tight muscle or press on a dense scar, take a moment to marvel at the incredible unseen physical dynamics happening right under your skin. Stay insanely curious, keep asking the deep questions, and we will catch you on the next deep dive.